Sorry, Don, it's JD here. So sorry to interrupt you. First of all, can I just say that your voice seems to be a little low. I don't know if anyone else is having that issue. If you could speak a bit closer to the mic for us.
Yeah, that's true.
Yeah, all right. So is that any better?
Yeah, that's much better. Thank you.
That's
I
great.
I need to hover over the mic, iPad. I had this morning. Okay. It's very rare that I'm accused of having a low voice, but occasionally it must happen. Right, anyway, so this is a dovetailing then of the final session of the conference and of the research showcase. So as it's probably better to be a keynote speaker, we'll stick to that. And it's a great pleasure to welcome Gary McLean from our School of Human Sciences. Gary's a specialist on viruses, and he's actually going to talk to us today about viruses and the hunt for a vaccine. And if you follow MetConnect, etc., he's been all over the news, from California to the southern tip of New Zealand and probably other places in between, and indeed maybe further afield than that if it's possible. So Gary, it's a great pleasure. The floor is all yours. We're really looking forward to hearing this.
Yes, great. Thanks very much, Don. Appreciate the kind words. Yep, we've been talking about viruses in this current pandemic for quite a while now, and yeah, United States, New Zealand, also UK, and well, it's affecting the entire world. So there's a good reason for that. I'd like to thank the organisers for the invitation to close the conference. It's really a great pleasure to do so. The conference, I think, has been great success. Lots of fantastic talks. Congratulations to all the speakers, and in fact all the winners that we just heard. Some of the student papers and posters were truly wonderful. And thank you to the audience for staying right till the end, or even just popping in now if you had to. So what I'm going to do now is I'll turn off my video for the remainder of the talk, the presentation, because I just got a message that popped up that my connection was a bit weak. So, and then we'll launch into the slides. Right, so as you can see, the topic of my my final talk for the conference is viruses and the hunt for vaccines. It looks like we're almost on the hunt for viruses as well, because there's so many different vaccine approaches these days, and that's really borne out by what's happening currently with SARS-CoV-2 and COVID-19. So the way I'm going to do this talk is broken into kind of four main parts. I'm going to introduce viruses and vaccines initially, and I apologize to the people who know plenty about this already, but I think for a general audience like this, it's important to get these key ground points known early on. And then I'm going to run through some of the work I've been doing over a number of years now, both in preclinical development of vaccines, and those that know this field will understand that this takes years and years, probably 10 years to go through preclinical and into clinical trials before you've got a functional and useful vaccine that goes into people. So there'll be two examples of that, the preclinical side and the clinical side, two different viruses, very different ends of the virus spectrum, one being very small and containing RNA, the other being very large and containing a DNA genome. And then at the end, because it's so current these days and it's affecting everyone's lives globally, we'll look at a little bit about SARS-CoV-2, what's happening with the pandemic, and of course, what everyone wants to know, will there be a vaccine for this particular virus? And you've all seen this image of SARS-CoV-2, and it's this wonderful structure which has got these spikes projecting out from the surface. These are, in fact, called the spike protein, and they are the critical part of this particular virus. So I'll move on now. Brief introduction. These key points about viruses are really important to understand. Viruses are so small, we can't see them with a normal microscope. You need a giant electron microscope. This is because they're measured in nanometers. So if you clench your fist and think of something that might be 1,000,000 times smaller than that, that's going to be about the size of a virus. Very, very small. These guys, they're intracellular parasites. They're not cells. They can't divide in the normal way that a cell would. They need to infect a cell, and that's when their replication machinery goes into overdrive, and they can actually do what they're supposed to do, which is very, very simple: make copies of themselves. But to do that, they have to hijack a cell. They have to infect a host, stay in that host. And obviously the host doesn't want that to happen because it's often damaging. But viruses have adapted over a long period of time to actually cause limited damage to their host, because you can imagine if there's no host, a parasite can't function. So viruses are no different than other parasites. They're very simple particles, shown here in this diagram at the bottom. At their simplest, they just contain a genome that can be either DNA or RNA. It's never both, it's one or the other. And that's in fact one of the ways that we characterize how different viruses are from each other. And all of the genetic information that makes the virus dictates what it does, which cells it infects, which host it infects, how it transmits, etc., is encoded in that genome. And they're very, very simple, very small genomes. They can't carry around too much information. But to protect that genome, they're surrounded by a protein coat or a shell that's called the capsid. And the proteins that are made up in that capsid are encoded by genes found in the genome. Some viruses also have an envelope around them. It's basically a lipid bilayer. It looks like what you might see in a normal cell, a mammalian cell. And often projecting from the surface of that envelope are further viral proteins that gain entry mechanism for the virus into cells. So the function of this entire unit is simply to stabilize the genome, to get that into cells so it can make copies of itself. That's simply all a virus will do. And this structure is really important for our immune responses as well. Obviously, we can't target viruses very easily when they're inside cells. Antibodies can't barrel through a cell membrane and attack a virus, but antibodies can do that when the virus is extracellular, when the virus is infectious. But we've also got these specialized T cells called cytotoxic T cells, or CD8 T cells. And what they're really good at doing is recognizing when a cell has been infected by a virus and then coming along and killing that cell. So the immune response we need to make to viruses is quite varied. We need antibodies to protect and stop viruses from getting into cells in the first place. But then we need these cytotoxic T cells to recognize and destroy the virally infected cells. So any vaccine will need to probably induce both of those components of immunity. So what are vaccines? What are the key points of vaccines? Well, the WHO calls a vaccine a biological preparation that improves immunity to a disease. And I think everyone will probably agree that vaccines are arguably the most effective health prevention mechanism that we've invented over time. They are thought to prevent at least 2.5 million deaths each year. And there are several criteria that are really important for vaccines in order to be useful and rolled out to the entire population. Number one, they must be safe. We can't inject a preparation into someone that's going to make a disease worse. It's got to be also very easy to administer. It can't be too difficult. Many people are frightened by needles, but that is actually a very easy way to administer a vaccine. These vaccines can't have adverse effects or very limited adverse effects. So all these clinical trials go through very long stage of development to make sure that vaccines are safe, cause any other problems, and they have to not cost too much as well. If you think you're going to make tens of millions of different vaccines to roll out to the entire population, it can't cost too much to do that. So large-scale application is critical in vaccine discovery. The preparations themselves need to last a long time. They need to be storable, and they shouldn't change as well. So they've got to be very stable preparations, because we don't want a vaccine for a virus, for example, reverting back to a wild type form. And very, very importantly, they must induce a protective immune response. And this is what we're going through now with the SARS-CoV-2 vaccine development. We're understanding, are these things safe? Can we scale them up to large scale to give to the entire population? Are they stable? And are they immunogenic? That is, do they produce antibodies and T cells and immunological memory, long-lasting memory, that when you do get exposed to the pathogen, your immune system remembers that and can fight it. They must also generate broad protection, because these pathogens, they change a lot, particularly viruses. They mutate, they evolve over time, and they render a vaccine somewhat ineffective over time. And that's time is very important as well. We need that initial administration of the vaccine to be quick and simple, but that immunological memory should last a long time. That's called longevity of immune response. So ideally, you get injected once with your vaccine, and that will give you lifelong protection. I don't think any vaccine really fulfills all these criteria, but we try and approximate it as best we can to get the best response. That number there in red, 17, that is the number of viral vaccines currently licensed to use in humans. So I'm sure you'll all agree there are many, many more viruses than 17. So we're a long way away from curing all viral infections. What are the different types of vaccines? Well, there are many that have been used and licensed for use in humans. I'll start off with the bacterial ones, toxoids. These bacterial cells, they produce a toxin such as tetanus or diphtheria. That's very easy. You can quickly inactivate the toxin, call it a toxoid then, and administer that as a vaccine. Conjugate vaccines are really for these bacteria that have these polysaccharide capsules around them. That polysaccharide sugar is not very immunogenic. The immune system doesn't react to it that well. So you often fuse that or conjugate that polysaccharide to a toxoid to improve its immunity. We can't do this for viruses because these aren't components of the viral capsid or its structure at all. So often what is used for viruses is an activation, so simply a chemical treatment to destroy the virus's ability to infect, or we can attenuate a virus. That means you can change the genome, passage the virus in cells so that it loses some of its virulence, becomes weaker, and we can use that as a vaccine as well. We can use small chunks of the virus, just a piece of the capsid. That's called a subunit vaccine. So these three approaches are the main ones used for viruses. And of course, the virus itself, because there are many, many different strains or types of virus, we can combine them into multivalent vaccines. So we can have several different viruses in one preparation, or a monovalent vaccine that is a single kind of vaccine. So lots of different approaches or platforms for making vaccines, but only some of them are actually that effective. And probably the best ones for a virus is an attenuated strain, because it kind of mimics the infection without being terrifically dangerous. So let's look at what happens in the UK. What do we get in our first year of life? Remember, when you're born as a neonate, your immune system is pretty bad. You're learning, you're reliant on the mother providing you with immunity during development and in those early years of life through breast milk. But there is a vaccine schedule mostly for bacteria, and I've highlighted the viruses in orange here. And you'll see it's really only there's two virus vaccines you get. One is polio. It's an inactivated polio vaccine, and you get multiple doses of that to generate long-lived protective immunity. And also in some regions of the UK, you're given by the oral route a live attenuated version of this gastroenteritis virus, rotavirus. In subsequent years, we have brought in a trivalent vaccine called MMR that we've heard a lot about. There was a doctor some years ago who linked this MMR vaccine, measles, mumps, rubella, to autism. It was subsequently discredited, and this has fueled a lot of the anti-vaccine movement that we're seeing today, people not wanting to take vaccines because they're deemed unsafe. Well, remember back, number one criteria for a vaccine is that it is safe, and these have gone through numerous years of trial and proof that they are indeed safe. So MMR is a great vaccine. It's a live attenuated trivalent vaccine. It's got 3 different viruses in it, and you get a couple of doses of that to induce your immunity. You'll also get boosters of polio, and then later on, in particular for teenage girls, human papillomavirus vaccine has been very successful in reducing the incidence of cervical cancer. As an adult, later on, you might receive an influenza vaccine and more doses of polio. So this is what occurs in the UK. There are many other viral vaccines that we don't routinely administer to people here, but maybe if you travel to certain regions of the world where there are endemic viral diseases, you may receive a different viral vaccine. So the whole concept with vaccines and how they protect you from infectious disease is this concept of herd immunity. So if you look on the left here, this diagram, we've got a herd of people. Many of them in blue are susceptible or not immune to an infection. But a couple of people, the red guys, come in here, they're carrying this virus, and they'll transmit it to all those susceptible people. And you'll end up with a population that's infected. Kind of what like is going on with SARS-CoV-2, because we've got no one that's immune at this stage. With immunization, what we're trying to do, excuse me, is turn those blue guys into these yellow guys that have been immunized, they're protected, and when these dangerous red guys come into the population, they won't be able to infect all those yellow guys, but the yellow guys will also, in the herd, protect all those ones that didn't get vaccinated, just because there's nowhere for the virus to easily transmit. You might see a small increase in the number of red guys or infected people, but the herd is protecting everyone. So the thing with the vaccine is you don't need to immunize everyone in a population. It's what we called the herd immunity threshold, and that number varies depending on the type of virus, the type of infection, how infectious it is. So it's been estimated for SARS-CoV-2 that we might need about 60 or 65% of the population to be immune in order to get herd immunity. For measles, we need a much higher number of people, 90, 95% of the population immune, because it's a much more infectious virus. So herd immunity threshold depends on how pathogenic and how transmissible the virus is in fact is. So let's go into now some of the preclinical development and the studies I've done over the last number of years. And one of the models I've been working on is rhinovirus, common cold virus. Everyone gets infected with this. It's a really annoying upper respiratory tract infection. It's not a killer, but in some people that have chronic diseases of the lungs, particularly asthma or COPD, the virus can travel down into the lung area and really cause very severe exacerbations of that underlying disease. It can actually be life-threatening in certain situations. So that's why we were interested in developing a vaccine for rhinoviruses, because it can be very dangerous. Now, this particular family of viruses makes developing a vaccine very, very difficult because there are at least 160 different strains or serotypes known. And these viruses are very, very small, only 30 nanometers in diameter. They don't have an envelope, so they're called naked. They have a small RNA genome, only 7,500 bases, so they've only got 8 or 9 different genes. 4 of those genes make up the capsid coat, and they're all the same. And this VP1, VP2, and VP3 protomer forms what is known as the capsid, and VP4 is buried underneath the surface. So antibodies can attack easily VP1, 2, and 3, but these tricky guys, they keep changing their surface. That's why they've got 160 different types. So antibodies to type 1 will not protect you against the other 159. And that's why we keep getting colds all the time. The other problem with rhinoviruses is their weapon is speed. Once you know you've got a cold or the symptoms appear, it's far too late. There's nothing you can do. So the only way to stop these guys is to have a vaccine that spans all of these 160 serotypes. So this is a classic problem and challenge with vaccine development. Here I've just shown the different types of rhinoviruses. They're grouped into A, B, and C based on their sequence of their genome, based on a particular region, and also they're characterized based on which receptor they use to enter cells. So all viruses need to attach to something on the surface of the cell to gain entry. That's called an entry receptor. And these guys use 3 different entry receptors. So there's evidence that the rhinoviruses are evolving quite a lot to use different entry receptors and create new strains all the time, which means we're always going to be getting colds. The capsid surface really is extremely variable amongst all of these different serotypes. And if you think about it, it's what the immune system can very easily see. That's where the antibodies will bind. And those are the guys that should be trying to protect you as well as the T cells as well. So what is the immunity to rhinoviruses? What do we know? Well, we know that the antibodies bind to the surface. Mostly they're exposed areas of the capsid. The problem is that this virus family has huge variability, and the neutralization by the viruses is serotype specific. So you're not protected against all the different viruses. The T cell responses, we know they're a big helper T cell response. It tends to be more directed to internal antigens of the virus, and these particular parts of the virus are more similar amongst the different family members. So there's more cross-serotypic reactivity with the T cells. But they're not the right kind of T cells. These T cells will help B cells make antibodies. They're good at that. But remember I said earlier on, what we need for these intracellular pathogens is this specific kind of cytotoxic T cell called a CD8 positive T cell that can seek out and destroy virally infected cells. Well, if you do a PubMed search for CD8 T cells and rhinovirus, you come up with basically nothing. There's nothing known. So it seems that these viruses don't really induce a very strong CD8 T cell response. And that can also be mirrored with SARS-CoV-2, we think, where there's probably very little information yet on that, but it wouldn't surprise me if there was a weak CD8 T cell response in this particular pandemic. The other problem with rhinoviruses is that the immune response itself is the trigger. It's what the symptoms are associated with the infection. So we're always faced with the same problem. Is the immunity you generate a good thing or a bad thing? And this is the problem all along with viruses and developing vaccines. So we're not the first people to try making a rhinovirus vaccine. It started way back in the 1960s, the Common Cold Unit in Salisbury. They did a simple experiment, inactivated a single serotype, gave that to people in their nose. What they found, perhaps not surprisingly, it didn't induce cross-serotype protection. So it didn't protect against many different types at all, only itself. But what they did define was that antibodies in your nose or nasal IgA, that's a specific class of antibody, that is a correlative protection. So if you make a good nasal IgA response to the virus, that will protect you against further episodes of that particular serotype. And they could last for about a year, which is okay, but it's not long term. Later in the 70s in America, they got a bit more complicated with the approach. They tried the multivalent inactivated vaccine approach. They put 10 different serotypes of the virus into people. And surprisingly, they showed minimal protective responses at all with this particular vaccine approach. So it was almost like the more viruses you combine into the vaccine, the less protective it became, which was very interesting. But at that point, rhinovirus vaccine was deemed impossible. Everyone gave up. And it wasn't until really, again, another decade in the 80s when different methods came along where you could immunize animals and make monoclonal antibodies that they started to understand a bit better the properties of antibody cross-reactivity. But the problem with this was that you couldn't infect these animals with rhinoviruses. They're human viruses. They don't infect mice. They don't infect rabbits. So protective qualities of this approach were really unknown. But more recently, perhaps in the 2010s and just before, some development of animal models of rhinovirus infection have been developed, and the vaccine has now been revisited, and that's where I came in. Our approach was to make a subunit vaccine. So remember back to the slide of the different types of vaccines. Some of these we can't do for viruses. We know that the inactivated monovalent failed. We know that the inactivated multivalent failed. We're really only left with two approaches. Attenuation, well, it's probably not appropriate for rhinoviruses because it's already kind of attenuated. It's not a very serious viral infection. So we went for a subunit vaccine approach. Remember, this is just a small chunk of the virus capsid, or just one antigen of the virus. And the theory we came up with to do this was, well, could we find a place somewhere in all those 160 different serotypes? There must be some areas that are shared. And if we can harness that area, inject that in as a vaccine, it might generate cross-serotype protection. And there was precedent for using a subunit vaccine approach for viruses. It's been proven to work for human papillomavirus, but then there were only 4 high-risk variants of the virus that we had to worry about, not 160. And hepatitis B, there's also a few serotypes and genotypes, but this actually has worked as well. So we thought this was a good place to start. And one of the first things we did was a bioinformatic approach. We lined up all the sequences of the different polyproteins, all the proteins made by the different serotypes of rhinovirus. We only used the type A and B for this because the type C were very, very new, and we didn't have all the sequence information. So when we did that, left to right, this is the entire genome or the polyprotein of all the different 100 serotypes. And this wavy line here it shows the similarity between all of those 100. So the higher it is, the more similar, up over 90%, almost approaching 100% here, and down in parts of the capsid, down below 70%. And these low similarity areas probably correspond to those surface exposed loops where the antibodies tend to attach, and that's why the antibodies are serotype specific. The other part of the virus polyprotein is all the enzymes it needs for inside cells to replicate. So antibodies to these regions aren't going to be very good, but these are good T cell epitopes. So we thought we'd first approach this area here called VP0, which is right at the end terminus. It's composed of 2 different capsid proteins. It has very, very high homology between these different 100 serotypes, and that was going to be our subunit vaccine. So first we had to produce that protein. And so what you do in this situation is you take the gene that encodes that region of the viral polyprotein. It's called VP0. It's made up of VP4 and VP2. That's the sequence, impossible to read in there, but that shows you how big it is, how many amino acids, its molecular weight, etc. And we use bacteria as factories for producing the protein. So we take the gene, we modify it in a certain way to make it express very well, pop that into bacteria, and they'll produce the protein for us. And then we have to purify that protein, inject it into animals, and look to see if they make the right kind of immune response. It's called immunogenicity. And we're looking for antibodies that will recognize that, and also T cells that will recognize that. Ultimately, what we need to do, if that preparation is immunogenic, is then do challenge studies. So that simply means you immunize and then challenge with the live virus. And since we'd developed our model, we could do that. So I can't go through all the data of how immunogenic our VP0 preparation was. This data I've summarized here on this slide, but it comes out of 2 or 3 different papers. And what we found with the antibodies was that, sure, we inject with VP0, this is mice, we can generate very good serum IgG, which is absolutely normal. That's what we'd expect. We also found IgA in the mouse lungs. And we found that these antibodies could bind to many different rhinovirus strains, and they were all specific for VP0. They didn't bind to other regions of the virus, which is completely normal. So remember, we're immunizing with one particular serotype strain that's highly conserved amongst all the different rhinoviruses, and it's generating a good spectrum of different antibodies that are binding to numerous rhinovirus strains. We also looked at T cells that were found in the lymph nodes of these animals. And these T cells were expanded in good numbers, and we found that they were producing this special cytokine called interferon gamma, which is really an antiviral molecule, and it's indicative of the right kind of Th1 immunity, which is specific or better designed for viral infections. And these cells would respond in the lab. We take them out of the animal and put them in culture, and they would respond by producing interferon gamma in response to many different rhinovirus strains, indicating that they were cross serotype reactive, and they were all specific for VP0 as well. So then we had to go on and test, okay, yeah, VP0 is immunogenic. What happens when we challenge these animals? Are they protected from rhinovirus infection? And the lab I was in, we'd published this paper just a year or so before. And what we could do, and we were the first in the world to do this, was we were able to infect mice with 3 different serotypes. I know 3 out of 160 is not much, but it was 3 more than anyone else could do. 3 serotypes intranasally, and induce very similar kind of pathology and events that would occur in a human. The virus could replicate, limited replication, but it still did replicate in the mouse cells and make copies of itself. We had all the same kind of inflammation. The innate immunity was similar. The adaptive immunity was also similar, all to what was occurring in humans. So we had a model that we could then test our vaccine in. So these were the kind of studies we had to set up, and asking the question, does immunization improve the outcomes following live virus challenge? So we could immunize and boost 3 weeks later with our vaccine. Remember, this is one particular serotype. It comes from rhinovirus type 16. And then we let these animals resolve for a while, and then we challenged them intranasally with a different serotype of rhinovirus. And we're looking for protective immunity through either T cells or antibodies. The questions we want to answer: does the immunization protect? Is it T cells, or is it neutralizing antibody that works better, or is it a combination of 2? And the really important one, are these antibody or T cell responses cross serotype reactive? We know they're probably going to work against type 16, but can we generate broader immunity? That's what we need for this family of viruses. So this is just one set of results that we found. So this is looking at the interferon gamma producing T cells coming out of the lymph nodes of these animals. And you just got to look at the black bars in each case. This is 2 separate experiments run 2-3 times each. It's representative of that. So the black bars are those animals that have been immunized and challenged. And you can see in all cases, there are more T cells producing interferon gamma in immunized and challenged animals. And along the bottom there, it's just showing what these cells are responding to produce their interferon gamma. So they're responding to specific VP0 peptides from different serotypes of rhinovirus, and the controls are very, very low. So we're generating good cross-reactive T cell responses in these animals from this immunization and challenge regimen. What about the antibodies? Well, they're also cross-reactive. I'm summarizing lots of data into a very small amount here just to show you a picture. Serum antibodies could bind to different serotypes of the virus, VP0 specific, of course. We could find these antibodies in serum and in lungs. And the antibodies, importantly, they actually neutralized the virus. So we do these assays in cells in the lab. We can infect the cells with the virus, the cells will die, and we can look for protection from death in the presence of these antibodies. And we found that IgG in the serum was a very, very good protector from this cytopathic effect of the virus. So the vaccine is looking like it's working quite well, but does it reduce the viral load, the number of viral particles that we could detect in the lungs of these animals? And the answer is yes, it does. It takes a little bit of time. Generally 4 to 6 days later is where we see effects. So the black bars are the ones that are immunized and challenged compared to the gray bars just challenged. And the black bars non-existent there at later time points, suggesting that that immunity we've generated is protective and is reducing the amount of virus in the viral lungs. So what are we going to do next? That's just a snapshot of the preclinical development of our rhinovirus vaccine. Obviously, we're going to improve our formulation. We're going to, with a subunit vaccine, it by itself, it's not very immunogenic. You need to formulate that with what is called an adjuvant. These are chemicals that are mixed together with an adjuvant to improve the immunogenicity and improve the broadness of the immune response received. And that takes a lot of work because there's very, very few adjuvants that are actually approved for use in human. So we're looking at developing new kind of adjuvants, perhaps in nanoparticles. We need to increase the breadth of our protection that we're seeing. That means we need to look at more rhinovirus strains, and that's currently being done right now. We need to define better exactly what the correlates of protection are. Is it just the neutralizing antibodies? Is it a combination with the T cells? Where do these need to be? Is having serum IgG good enough, or do you need nasal or lung IgA? Where should those T cells be? What is the phenotype of those T cells? You'll notice I didn't talk at all about CD8 T cells. I was just mentioning T cells in general. So again, we're not inducing great CD8 T cell response with this particular vaccine, and maybe that's something that we do need to protect against rhinovirus. And we're also looking deeper molecular studies of the exact epitopes found in the vaccine preparation so we can minimize the amount of the vaccine that we're giving. These are all ongoing studies. Ultimately, we want to get this into people. So we're at a stage where we're answering many of these, and that will be the next step, is to go into clinical trials in people. But it takes a very, very long time to get through this process. Most of these data we've taken out of a series of papers that we've published over the years. So the next model system I've been working on is something that's been in human clinical trial over a number of years now, and that's a herpes virus called human cytomegalovirus. This is completely different to rhinovirus. It's a big DNA virus. It's got a genome. It's got about 200 different genes in it compared to 8 or 9 for rhinoviruses. It's got an envelope around it, unlike rhinoviruses, which are naked. It's got a DNA genome instead of an RNA genome. Most people are infected. It infects lots of different cell types, so it's not a respiratory virus. It can infect almost any organ and cell in your body. Mostly it exists. It's not a big problem for healthy adults, so it exists subclinic. But it can be very, very serious for those people that have weakened immune systems and can cause pretty severe disease in people receiving transplants. Remember, they're on immunosuppressive therapies. People with HIV whose immune system is damaged have a real problem with HCMV. And probably the most important area of health for this virus is congenital infection in pregnant women, where the virus is transmitted to the developing fetus and can cause quite severe developmental abnormalities in the neonates. So the Institute of Medicine in the United States has categorized this as a priority 1 vaccine candidate. So there's been many, many studies looking at different vaccine approaches. And the main approaches that have been used are the classic attenuated viral vaccine. This is going to generate good, strong T cell immunity, maybe not so good antibody immunity. But this has been in people many, many times. It showed some efficacy against CMV disease in transplant recipients, but it doesn't prevent infection. It seemed to be very weakly protective in a human challenge, and it failed to protect pregnant women or women of childbearing age. So other approaches have been undertaken with this, and this again is back to the subunit vaccine approach. And one particular glycoprotein, surface glycoprotein of the virus called GB, has been used quite extensively, and it's combined with this remember that chemical composition that improves the ability of vaccines to work, called MF59. So GB MF59 has been in people lots of times. It's immunogenic in people that have already been exposed to the virus, so it can boost the already pre-existing immunity, which is good. And it shows efficacy protecting against CMV in seronegative women, and also 50% efficacy in controlling episodes where the virus flares up in these people receiving organ transplants. So I have been interested in HCMV GB for a number of years now, and this molecule has been very well characterized, characterizing the various epitopes and regions on the molecule for what is important for immunity. And my favorite part of it is epitope is called AD2, which was found on the tip of the spike up here, this orange part of the GB. So that would be the outer part of the virus, and then down here at the blue region would be the viral envelope. This is protruding from the surface of the virus. So it's right exposed out there on the top. So you can imagine antibodies would like to stick to that area and can in fact neutralize the virus by doing that, because glycoprotein B is critical in getting the virus into cells. And we've solved structures of antibodies binding to AD2 in the past. The problem with AD2 is that not everyone makes good antibodies to it, and it's really unknown as to why. But if you do make antibodies to it, you've got a good neutralizing immunity to that virus. So the aim of a vaccine, in my opinion, would be to generate good neutralizing antibodies to AD2. So we looked in one of the clinical trials exactly what kind of antibodies were being made in response to the GB vaccine. And we're looking here at seropositive people on the top and seronegative people. And the first thing you'll notice is that the vaccine itself really induces a very good antibody response. This is IgG to GB. In those seropositive individuals, if we look at the AD2 response, we see that there's a variable response. Some people don't make antibodies, others do. And the vaccine always boosts those antibodies to AD2 in those people that already have them. But if you don't have antibodies to AD2, if you're seronegative, in fact, for all GB antibodies, the vaccine is not very good at inducing antibodies to AD2. And the same actual picture appears for IgA as well. No one's really looked much at IgA responses to HCMV, and we see very, very similar results to what we observed with IgG. We do get a boosting of the GB response in seropositive people, a weak boost to AD2 as well, but in seronegative people who have never seen the virus before, yes, the vaccine makes good response to GB, but nothing at all to AD2. So we're trying to understand why this vaccine isn't inducing those particular antibodies. We went further and looked in people that are receiving solid organ transplants and getting new kidneys and livers, and we showed that the AD2 response is boosted with the vaccine, and it lasts over a good period of time, up to 7 months. In fact, if you take away all of those AD2 negative people out of the picture, you can see there's a really good AD2 boosting antibody response in these people. And we looked even deeper. I know this is really hard to see this one, but we're comparing 4 different epitopes of GB, and the one in the red box is AD2. It's the only one that showed any correlation at all with protection against viremia. So this is when people were then exposed naturally to virus. And we just count up the red dots if you can. You can see here, and those that received the vaccine and boosted their AD2 response, there are less red dots, so there's less viremia episodes. So that's a correlated protection for this particular vaccine if you can induce AD2 specific IgG. So we're going to go on further with this GB vaccine trial. We're going to look deeper at the IgA responses because we think this might be important, mucosal immunity because the virus is transmitted by that route. And I've been going on about this for years now, and perhaps a better approach for this particular vaccine is to first prime with AD2, induce that antibody response, and then use GB as your booster, and that will generate a more broad immune response. So that's where we're at with HCMV. That again, these data come picked out of many different papers we've published over the years. So I just want to finish off now with what's going on with SARS-CoV-2. This is impacting on everybody's lives. You know, we all know it's a coronavirus. It's just appeared recently in China. And in fact, it was really only discovered in late December last year, this unknown pneumonia in people. And so fast to generate the viral genome sequence. January the 10th, this was sequenced and shared before the virus even had this name of SARS-CoV-2. It had a different name. And the genome itself of coronavirus is really interesting. It's a big RNA virus, it's got 30,000 bases. It's got these different components to it. The spike is what makes those red features on the surface and gives the virus its name, coronavirus. The spike is critical for getting the virus into cells. We know the entry receptor is ACE2. If you slice through the virus in this cartoon and you see the picture there, you see it's got a membrane, it's got its RNA in the center, it's got 4 different structural proteins, nucleocapsid, spike membrane, and the envelope protein as well. And human coronaviruses, not many of them that infect us, there's only 7. This newest one, written here, had its original name, is closest to a bat coronavirus. In fact, it must have come from bats. And it's very similar as well, sequence-wise, to the original SARS coronavirus from 17 years ago. And there are several other different coronaviruses that really cause mild common cold-like disease. And also we've got this MERS coronavirus, which is transmitted from camels to humans. So we know with this particular pandemic, 4 out of every 5 people, the infection results in very minor or even no symptoms. The virus transmits very well. It's got this reproductive rate of around 3 or 4. So on average, 3 or 4 people will be infected from one person. There's evidence for asymptomatic transmission, and we know the case fatality rate can be approaching 1% in certain areas. And this is the typical pattern of what happens. There might be no symptoms for up to 2 weeks, then you'll develop a fever and a cough, breathlessness, you might be hospitalized, and you'll either die or recover from this. And there's huge variability in the way people respond to this particular virus. So I just picked out these numbers from yesterday. This morning they're confirmed from yesterday. There's been over 13 million lab confirmed cases, over 500,000 deaths. These numbers are increasing all the time. There's probably much higher numbers of cases, but they're not all laboratory confirmed. And the vaccine landscape has really gone crazy over the last little while. It's unprecedented how fast this is. There's now 25 different candidates in the clinic and over 130 in preclinical development. And some of these really gone so fast since the discovery of the virus in just 3-4 months. And there's a number of different companies, different approaches using many different platforms for vaccine. We've got inactivated, we've got the use of a non-replicating vector to put the spike gene in. They all tend to be focused on generating antibodies to the spike, which we know are protective for this particular virus. And many of these might actually make it through to be an actual vaccine, perhaps as early as the start of 2021. According to Dr. Fauci, the director of the NIAID, who claims that we might at least know whether or not some of these vaccine approaches are actually going to work, but it's still days. There's a lot of questions about COVID-19, mysteries. Why do people respond so differently? There's lots of thoughts. Is it genetics, general health, age? Is it the immune response you make? What is the immunity and how long does it last? We still don't know the answer to all this. Neutralizing antibodies exist. We don't know much about T cells yet. There's some evidence that these antibodies disappear after 3 months. So what does this mean for a vaccine? Will we ever get a vaccine? Will we escape from this lockdown? Will the virus change? Has it developed any worrying mutations? And there's evidence the virus is mutating, maybe subtle, but there could be one in the spike that is taking over called D614G. So there'll be more papers on that. Will a vaccine work? I mean, there are no guarantees at the moment. We have to hope there will be one. I think the advantage we've got is a lot of work going on in this area. And where did the virus come from? Well, we know it came from bats. Did it come via an intermediate animal? We still don't know because we haven't found yet a coronavirus that's very close in an intermediate animal. So I think would like to finish here with what will happen next. How are we going to solve this problem? I like this diagram because it shows over time what's happening in the different continents with the virus. We know original spike in China here, in Asia, and now this rise up being fueled mostly by India. We've seen the peak in Europe that is dropping away. We've seen America have a peak and now a second peak as well. The Middle East is rumbling along as well. South America, really the hotspot at the moment. Africa probably yet to see huge numbers of virus. So what's going to happen when kids go back to school in September, October? The public compliance with lockdown and behaving appropriately, wearing face masks in shops, etc. Will the virus mutate even more? So will we have a vaccine to solve all this? Unlikely, but there's a lot of work going into it. I think I'll stop there. I've gone far too long. Sorry about that, but lots of people that helped out with much of this work and will do so in the future. Thank you.
Well, thank you very much, Gary. We can indulge you the extra time because it's really fascinating, and I think you pitched it just right for a pretend scientist like me to understand it. It was really good. Thank you. Lots of applause in the timeline. Does anybody want to get a question? Anyone's hand going up? Lots of comments. Oh, there's a hand. Oh, Una, the floor's yours. Turn your mic on and go.
Hi Gary, thanks for that. Really good talk.
You're welcome, Una.
What do you think is the basis of the non-antibody mediated immune response to coronavirus?
There's going to be a certain amount of innate protection. I think it depends on your inflammatory response, exactly how you respond to all these stimuli that the virus is providing. So your innate immune system senses there's something wrong and responds in a certain way. It's whether you overreact or not to that, which will generate the severity of an infection. Of course, I think you still need adaptive immunity to clear out the infection over time. And it's probably going to be a CD8 T cell response.
And do you think that's the basis of the severity?
I think the inflammatory response that's sensed from the innate immune system is triggering a lot of the severity of the disease, yes. But I think it's too soon to know exactly what the immune correlate of protection will be for clearance, but it's likely to be T cell as well as antibody.
And what do you think the basis of
last one?
Sorry, what do you think the basis of children not responding, or children responding well, children not responding poorly is?
Well, that's a really tricky one because I think maybe their immune systems are less experienced and less overreactive than an adult's one is. There may be some component of their innate immunity which is actually better at protecting early on, and they don't generate the high levels of virus replication. It's not just a respiratory virus, remember, then it spreads to other parts of the body, and there's a big component of thrombosis in the cardiovascular system as well. There's just a myriad of different symptoms, and maybe children are just generally more healthy than adults because they've been exposed to less junk over a period of time.
Thank you both. Now, Klaus has resisted the temptation to mention something I read about, that Germans have more dark matter, and this is why they have escaped more likely than us. But he never mentioned that. What he's actually asked, Klaus has, is, oh, he's disappeared, what did it feel like when the antibodies expected did not materialize?
Which antibodies are we referring to here?
I think he's referring to perhaps one of the experiments you talked about, maybe. I don't know. Well, if we don't know, we'll move on because Lee Jun.
No. I think it might be the HCMV ones, the 82. Okay. Yeah, I think that's a problem with this virus in that it has a lot of ways of tricking the immune system. It's the big genome. It carries around these genes to avoid immune detection, and other viruses don't do that. Their weapon is speed, but a big lumbering herpes virus has to find other tricks. And it carries around these things, and it tricks the immune system, saying, don't do this, do that, don't do this. And maybe one of the mechanisms is don't make antibodies to that area because it knows it's vulnerable.
Excellent. I think he was asking that sort of question, though, that we all wonder, scientists try and try, don't they? I mean, this vaccine thing must be resulting in kind of daily disappointment, you know. In fact, even the way it's reported superficially in the press for people who aren't scientists, you think, you know, it's moving too quickly. Antibodies don't last long enough. I think it's really interesting. It's a tough world, but it's probably even worse to try and think about how you might plan a journey to Pluto that we were talking about at the beginning. But it's perhaps in the ballpark for inner space. Anyway, enough of me. Lee Jun would like to answer a question. Turn on your mic, Lee Jun, and the floor is yours.
Okay, thank you very much, Don. Excellent talk, Gary. And I don't want to ask any further scientific questions. I just want to get some information about, do you have any work on the structure of the virus and what the methodology, you know, is possible in London Med we can do over here? Because I have small collaboration work with Chinese colleague. We're doing some modeling on these antibodies stuff. So do we have this information? Yeah, that's my only question.
No, thank you, Lee Jun. Yes, I think what we can do in our lab is we could work with a pseudovirus. That's a different virus where they splice in the spike gene from SARS-2, so it's not dangerous, and that can be used to model certain parts of it. I know it's not going to be perfect for the other structural components of the virus, but it will be good enough for the spike. And we can also work with one of those endemic coronaviruses that causes a common cold. And there's one in particular called NL63, which uses the same entry receptor as SARS-CoV-2. So that would be a good approximation that's not going to cause a horrible pandemic, and it's a level 2 pathogen. I hope that answers your question.
Okay, thank you. We can discuss this further when we meet.
Yes, indeed.
Good idea. And a question from Jamil in Ireland. There's a couple in the chat session, which I'll ask in a minute. But Jamil.
Thank you, Don. Thank you, Gary. It was a lovely talk, and especially for a closet virologist like myself. I was wondering whether, you know, just a comment and a couple of little questions about the promise of the vaccine against this SARS-CoV-2. I mean, compared to flu viruses, coronaviruses don't have as many ways of interacting with cells. So if you can block that interaction, one would hope that, you know, you could block replication and be a chance for the vaccine to be effective. That's just a comment. But what I was really wondering about was the development of a vaccine when it comes. And for example, these approaches using DNA plasmid and RNA vaccines, the scaling up of this it's never been done. You know,
Yeah.
they've never scaled up to hundreds of millions, and
Yeah.
I was wondering if you could comment on how difficult that will be, and also with regard to safety of testing vaccines. You know, normally you test it in one animal and then test it in people after that, so you know if there's side effects and so on. But now we're testing it in multiple animals and a few people at the same time. I'm just wondering if that's, you know, obviously we're doing this to speed things up, but there's a risk element as well. So I wonder if you could comment on that, please.
Yep, thanks, Jamil. Yeah, totally agree. The speed of this is unprecedented, and it's really is a bit of a worry, to be honest. These vaccine approaches are unproven. Many of them, the DNA vaccine or the RNA vaccine, is difficult to know how they're going to scale up. But at least it's a bit easier than having to scale up a subunit vaccine, which, you know, all the hard work you have to do in the lab. But RNA and DNA, at least it's a bit simpler to make. But I agree, making hundreds of millions of doses is not been done before. And the safety aspect, again, is another one. We don't know what's going to happen with these DNA vaccines when they go into a huge number of people. And I think number one priority still has to be safety. Even if they're rushing to get this done, they can't compromise on safety. So that's what's going to make this process go longer. And the vaccine probably not ready until late next year.
Yeah. Fantastic. Thanks, Gary. And I'll catch up with some rhinovirus questions when we're back in the office.
I hope I can see you in the office soon, Jamil. Yeah.
Thanks, Gary.
Brilliant, Jess. Listen, there's a couple of student questions. I don't want to let them go. So, Wally, would you like to turn your mic and ask your question? I presume it's your BAME question from the chat line. And while he's turning on, a yes or no answer from Christopher Rawlings. Is Patrick Vallance right to predict a second wave with 120,000 or 125,000 deaths as a worst case scenario? Seeing as half of that number have actually died in the UK already, as if there was a second wave, the answer is yes. But what do you think, Gary?
Yeah, it would not surprise me at all. If you go out now, you'd think the general public thinks that this has gone away. This has definitely not gone away, and you just got to look at what's going on in America. It's extremely frightening. Florida, California, Texas, Arizona. There's a definite second wave there. Yeah, I'm worried that this public compliance to this hasn't gone away at all.
No, I agree. And the real data that you'll see next year about the actual excess deaths, it's already at half that number anyway now, in actual fact, because they record 45,000 deaths, which we know is nowhere near anyway. Wally, sorry to keep you waiting. One last question from Wally, then we're going to...
Yeah, it's actually on the chart, but being originally from Central Africa, where the virus has been managed quite well, how come BAME are said to be more vulnerable to COVID-19? What's the rationale behind this claim?
Yes, well, I mean, that's been well established, that the BAME community tends to respond more severely, and I think that comes down to, must be some genetic component of the response to the virus. I don't think there's anything in concrete yet that we can say that it's X is causing that and Y, but it'll take time for these studies to come out, and I really cross my fingers and hope that the virus doesn't take hold too badly in Africa, because it could be quite devastating, because we know what other virus epidemics can do there. And so jury's still out. We don't know, but those studies will be ongoing, and I'm predicting some kind of genetic components in innate or inflammatory immune responses.
There are lots of questions. I guess we'll have to pick those up in as part of the recording. I understand now we're staying in this session and switching to the final conclusions for the conference. Would that be with you, Klaus?
Yes, indeed. And Christine, are you
I thought
there?
you'd be comparing. Are you doing it together? Are you at the same time?
Yes, I'm here.
Yeah,
yes.
I just wasn't sure if are you doing a little slot. Anyway, I'll step aside then and let Klaus and Christine take over. Thank you all very much. Excellent talk, Gary, and an excellent discussion. Some great questions down here, which we just couldn't get to them all. But never mind. We'll have to have another one on this. You can update us later on. Thank you all.
Definitely. Yeah. Thank you very much for everyone for listening. Great question.
Hi, Christine.
Thanks, Gary. It was great.
Who goes first, Klaus?
Well, do you want to go first? Shall I? How shall we do that?
I could get this over with for me, but
Okay,
yes,
you go
that would
first.
be great if I could.
Okay.
Okay. So, it's my privilege to make some concluding remarks from a research student perspective. The conference has been fantastic, perhaps a turning point for research across the whole university, and the culmination of goodwill, hard work, and imagination. The success of this conference has been a massive team effort, but I would like to pay tribute in particular to the vision, dedication, and energy of Klaus, Rena, Chris Rawlins, who is student co-chair of the Student and Staff Liaison Forum, and Myra Gallardo, who is president of the Postgraduate Research Society, and she has brought together and nurtured a large and highly supportive student research community. And in fact, the WhatsApp group is going wild at the moment with positive comments such as, "Congratulations to everyone who prepared, presented and attended. This was a very good experience." Personally, I'm taking away many things conceptually from this conference, summed up metaphorically by yesterday's opening keynote lecture on developmental psychology and spatial binding. How do we make sense of the dots within the spaces? The conference has generated significant opportunity and excitement about the future. The interdisciplinary vision that underpins the conference has been a showcase for London Met's unique assets as a place for research-based study, building on the university's global perspective and commitment to social justice within diversity, its understanding of tradition and willingness to explore and experiment. In the future, I'm sure the conference will be seen as representing renewed entrepreneurial spirit, confidence, energy, and momentum. It has also strengthened the community of students and staff who are passionate about research. A truly collaborative space in which everyone benefits from everyone else's success. This level of engagement can be further strengthened, perhaps drawing on the ideas of co-creation discussed in the presentation on design thinking. New generations of researchers, and I include myself, not by age, but by life trajectory, are being formed in this crucible. To borrow the words of philosopher Hannah Arendt and substituting the word students for children, education is where we decide whether we love our students enough not to expel them from our world and leave them to their own devices, nor to strike from their hands their chance of undertaking something new, something unforeseen by us, but to prepare them in advance for the task of renewing a common world.
Thank you, Christine. Very wise words, and really one could leave it here. But I should add my few words. I mean, first of all, I'm terribly grateful that the conference has really happened without major hitches. We had our share of problems, but overall it was delivered quite well, even though it was prepared just in time. I should share my video that you can see me. So my overwhelming feeling at the moment is really relief and gratitude. A very big thank you goes, of course, to the RPO team, to Maeva and Ollie, and a very big thank you also to the CPA team, Digby, Sue, Janet, JD, and Wilco, you know, for their excellent work before and during the conference. The biggest thank you, in my view, goes to all the presenters for sharing their wonderful research and by and large keeping to time. I mean, I'm surprised that I was involved in the organization of something that is nearly on time. It's 12 minutes past 5, so I still have a few minutes to go on. We had a conference topic between tradition and innovation. It was evident, of course, in Don's wide-ranging lecture this morning. He emphasized that we need both, of course, tradition and innovation. You need to know about the tradition to be innovative. Also that perhaps it's a wrong dichotomy. Also that perhaps today's innovation might become tomorrow's tradition. So my very superficial impression was that most papers were really rooted quite firmly in education. My impression was that about the science, the ICT papers. But of course, within that tradition, you can innovate and create new ideas. The first keynote was referred constantly to the tradition of research it was based on, but then found an interesting difference between ASD and ADHD children in spatial binding, and also made a recommendation for practice, how actually that could make something better in the world. Some papers try to rock the boat a little bit, and I come back to Alicia Knast's paper, where really the title might have suggested that one would hear a heartwarming story about the tradition, the particularly Polish tradition of boat instrument making. But at the end of the paper, the carpet was pulled, and the outcome was really there's a European tradition. There is no particular Polish tradition. Now, that is, of course, a paper that says one tradition here is kind of misconstructed. And so it's a brave outcome of archival research, of going back to the sources, looking, is there really a basis for claiming that? And of course, the paper about the Persian carpets also rocked the boat in suggesting that the VNA interpretations might have to be amended. Today we heard a paper by Bartholomew Beben that kind of wants to enter new territory, researching really on online communication and looking at the compressibility of the texts that are delivered there, and seeing whether what has been done with Bible translations, for example, where huge differences were found, how compressible the data was, whether that also applies to, online communication, and whether these differences hold. And of course, if you do something where you might come out with data that might surprise some. In linguistics for a long time, we had sort of the idea that all languages are equally complex, and if one language is more complex in one area, it is less complex in another, and it all works out fine. But from the linguistics perspective, we have thought that that is thoroughly wrong, and there are huge differences in complexity, which do not, I should emphasize, translate into differences in what you can communicate with these languages. And so one must be very careful as well in communicating research outcomes, that one keeps a little bit control of the interpretation, what is done with your outcomes, your results. And I think there was, in our research papers, we heard in some of them was perhaps a little bit of dynamite as well, and that's a good thing, and needs to be considered. There were other topics that are important. A lot of the papers had a practical application. Some actually set out to improve some practice, the teaching practice through the use of drama, or but others, also, had to improve how counseling happens, what subconscious messages are conveyed, how people feel excluded or feel victimized in counseling. A subtle approach, a very scientific approach to a problem by Amy Beddoes, very much to be recommended. So we saw some marvelous research. Other topics were certainly the co-construction of knowledge in relation to teaching design, but also the co-construction of identity through Pidgin and Menu United. So West African identity through Pidgin English, and to research that, how that is done in online fan clubs, in the speech there, innovative and new land entered. We talked about the shared language. Yes, a very important. Listening to some of the science papers, I wished I had more shared language with the sciences, but some papers actually translated it quite well to me. Of course, Gary McLean's keynote, but also some other papers which really could be quite well followed, and even in a lightning talk, a few minutes, but Marjan Jedi managed very well to make me understand how the cells change in the immune system. Well, I don't want to go on for too long. We had applications AI. I mean, that was also something I did not know how strong that is. Really, the research, the very much alive doctoral community, doctoral student community, and I should share my observation that actually the science and the ICT papers tended to acknowledge the supervisors, and also some of them mentioned research ethics, and overall they came over, quite a tight community that is there, and just as an observation, and perhaps something we can learn from that. That is as much as I should talk about these slight mishaps. I think there was also, as a feature of the conference, some great innovation, a great improvisation. Improvisation. When the opening note couldn't be given by Don stepped in. When a speaker, Peter Newby, could not give his paper because he was in A&E, Anmaki stepped in, and when Chris Rawlins, as a co-chair, did not have any internet access, Mignon Cristofoli stepped in and did a marvelous job of co-chairing. So overall, I wholeheartedly agree that this could be the start of, a tradition of a new research culture, of an inclusive research culture with students, and the kind of co-preparation that we did. I think we should strengthen for next year and see that we can emulate the Teaching Learning Conference in establishing a tradition. And we are now 6 minutes over, but we started a little bit late. Before we all go away, I would like to invite all of you to share your thoughts, to say what you observed, and offer your thoughts on the conference and also on the future. But before I do so, I should mention that we would like to collect the contributions in writing and publish them as a London Met publication in some form. And we will write to you about this in due course. So don't forget what you have said. Keep your manuscripts or your notes because we very much would like a written contribution from you for a publication, at least an online publication, and we have to discuss whether we can go beyond that. So from me, a very, very big thank you to London Met for hosting that, and to everybody for participating and being so supportive and indulging us organizers a little bit because it was a new ground for us, and behind the scenes there were some frantic things happening. Thank you very much. So any contributions? Any questions, any thoughts to share? I see Chris says something positive, great conference. Thank you, Chris.
I have something to say, if that's okay. Sorry, everyone. Good afternoon. On behalf of the Postgraduate Research Society, I believe that it's been a really great experience, and I think this is something we've been waiting for. And then I really want to thank London Metropolitan University, and I'd like to thank the research office and everyone who's been involved and volunteered. And I'm really proud of each and every one, and I want to say thank you very much, Klaus, Dawn, everyone who's behind it, and yes, well done. Thank you very much.
Klaus, I'll jump in if you don't mind. Klaus, you are our leader, our head of graduate school. We've thanked everyone many times, and they all deserve all those thanks, but I want to thank you personally with nobody else attached. You've done a great job. I know you've done it's been a big job. You've done a bigger job than just organise this conference. It seems like a really fitting end to an academic year when you've really been a driving force. And from me personally, and I know on behalf of everyone else, thank you so much. That seems like a good place to end.
Yeah, well, thank you for your kind words, Don, and thanks for your support in organising it all. I mean, that was great.
It's just a testament to everybody, isn't it?
Shall we? Okay. Is there a raised hand?
Yes, Rena.
Hello, everyone. Yes, yeah, just to say a couple of things, my observations. I think it was, we had COVID-19. We couldn't be all together in a room, but I think it worked really nice, this setting, to have this conference over, with the internet connections and everything. And it was really nice to see that we have participation or, attendees from the Balkans, Oman, Iran, Cyprus. So maybe it is, it can be an idea that we can explore in the future, so we can open this research meeting to everyone, really. So yes, and thank you very much for giving us the opportunity to organize such a great event. Thank you.
Thank you as well, Rena. Right, should we say it's closed, Klaus?
Yes. I think everybody should go out, enjoy themselves if they can, or enjoy themselves in their home where I'm sure it's safer. And let's meet at the latest next year again. And I hope in situ rather than online.
Fingers crossed. Bye, everybody.
Bye, everybody.
Keep safe.
I shall stop the recording now, Klaus.